I would like to introduce our first speaker, Dr. Arnaud Martin. Dr. Martin is an assistant professor at the George Washington University in Washington, D.C. He earned his master's degree in cell and developmental biology in France at the École Normale Supérieure de Lyon. Dr. Martin has worked on the genetic basis of color pattern formation for the past 15 years, which was divided between earning a PhD in biological sciences under Dr. Bob Reed at UC Irvine, a split postdoc at Cornell University and UC Irvine under both Dr. Reed and Dr. Tom Schilling, and a final postdoc with Dr. Nipam Patel at UC Berkeley. Dr. Martin's research focuses on deciphering the developmental mechanisms that make butterfly wings a crucible of morphological diversity. He is also sponsored by the NSF to establish the pantry meal moth as a laboratory system for studying lepidopteran insects. He has 9 years of experience applying CRISPR techniques to non-model organisms, integrating the gene phenotype relationships with DNA editing experiments that tweak body region identity, the Hox genes, the morphogenic signaling and pattern formation, the Wnt pathway, or the identity of iridescent ultraviolet colors. He's also developed a laboratory course where undergrad students generate butterfly wing mutants in the classroom, from experimental design to embryo microinjection of the CRISPR reagents. Ladies and gentlemen, Dr. Martin.
All right, thanks so much to the GES organizing committee. So quick for the technical side, do you have the right part of, or the top part of the screen ghosted by my panel? Can you see the whole slide right now? And can you hear me okay? Yes, Brendan? Okay, good. And so you can see the top, it's not shadowed by anything, right? All right, sounds great. All right, so today I am gonna talk about my ongoing.
Dr. Martin.
Can you say again? All set, all good?
All good.
Thank you. So I want to showcase a little bit the work I've done since 2014 on various non-traditional model organisms to study the evolution of development. So while I will talk a lot about butterfly wing color patterns, which has been really the bulk of my interest, I will also mention a little bit of work on crustacean limbs that I did as a postdoc. And then if I have time, I will talk about the use of CRISPR technology in the classroom to give undergrads hands-on experience with genome editing. There is my Twitter handle here, and don't hesitate to take screenshots or photos or whatever you want and share on social media. There is no need for secrecy on anything here. All right, so my big question, my big interest is to interest how organisms, how complex biology has been tinkered during evolutionary time. So as an example of evolutionary tinkering, you can read this beautiful quote by François Jacob here. Complexity in biology, complex structures don't come out of nowhere. Usually they evolve by modification of pre-existing structures. And as an example, the extraordinary complexity of the ossicles that allow us to listen to sounds, or the product of a complex history of modification of small bones and ossicles at the base of our jaw in our proto-reptilian ancestors. And I'm really fascinated by this kind of situation where you have pre-existing structures that are tinkered, that are modified over the course of mutations and selections, and that acquire new function over time. And I will continue a little bit this analogy using an anthropomorphism. So if you think at this, case, hypothetical case study, the evolution of utensils, maybe you have ancestral utensils with simple building rules and simple functions. And by combining their features, you can make more diversity, right? And if I can give you a speculative example of that, maybe when you look at this cockatoo bird, it is speculated that the crest of this bird originated by the co-option, so the recycling or redeployment of developmental information that is typical to the wing. which gives this articulation of the feathers, and those feathers are morphologically close to the feathers of the wing, and they're not typical head feathers, right? So maybe by tinkering their developmental program, if I can say, modifications of the genome in particular genes that are good at giving instructions to development, you can maybe position wing feathers on the head of this bird. So I'm interested into this kind of question. And so first I'm gonna mention this kind of study in a little crustacean that I studied starting in 2014 when joining the lab of Nipam Patel. So this is Pariale, it's a small crustacean. It looks like a shrimp, it's not really a shrimp, it's more like a beach hopper or a sand hopper. And it has an arrangement of segments like any arthropod, and it has 11 types of appendage extension. So those are all types of legs, but they are specialized into sensory structures like antennae, feeding structures in the mouth or the claws for food processing, different types of legs, and as well as propelling mechanisms or little forks or anchors to live in an environment with a lot of water movement. So we like to compare them as a Swiss Army knife of different tools, depending on the function and morphology of those appendages. And those appendages need to acquire the right identity across the antero-posterior axis. And so to study the development of those organisms, there was not much available before 2014. There was a little bit of RNA interference, no transgenics, actually, yeah, a little bit of transgenics, but it was very troublesome, I would say, to study gene function in this organism. And so 2012, 2013, the CRISPR papers start to come up. So we started to use CRISPR in those non-model organisms, and it worked beautifully. So to be very, very clear, we used genome editing as a perturbation assay. We have not done a lot of gene insertion or precise editing. Rather, what we do is that we inject Cas9 as a protein with a guide RNA. So the two things form a duplex, which you can see in the background. And we deliver them in the zygote, so in the first cell of this organism. And we hope this guided missile to go into the genome and cleave the DNA at that target, in that specific target gene. And because DNA breaks have to repair to allow the cell to continue with its cell cycle, it usually goes under a process of repair that is error-prone, called non-homology end joining, and we induce frameshift mutations in the genome at our specific target. So it works a little bit like this. This is a former version of Cas9 here. It's the zinc finger nuclease. You can see it on this cartoon of a wrestler, and it's really destructing the DNA, right? It's cleaving your DNA. It's a destructive assay that you're doing, and we induce in a targeted manner frameshift mutations in protein coding exons, and that's how we remove genes. So it's not really genome editing. It's more interrogating the genome by removing one variable, one gene at a time. So I want to talk a little bit about the development of this crustacean, Pariale. It has a very elegant development because it has a slow cell divisions and holoblastic cleavage. Holoblastic meaning that each forms cells of the same size. So we could inject the zygote right after fertilization here, and we did it, but another sophisticated version of this is to inject just one cell at the 2-cell stage, so just one side. And when you do that, you generate an organism that is half mutant, left and right. So it's going to be mutated on just one side, and you can compare the left and right of those animals. And I'm showing you here an embryo that is stained for a Hox gene called antennapedia. So on the right side, you have the side that is not injected for CRISPR. You can detect the antennapedia protein. On your left, you have the side that is mutated. Antennapedia as a protein is not present in the side of the embryo. We successfully removed it because we induce little DNA breaks, exactly 3 nucleotides upstream of the PAM sequence. Here, the guide RNA and the PAM sequence are in reverse orientation. So about right here, just after the letters CAT. And the DNA has to repair, and very, very often it will induce those little insertions or those little deletions. And so the number of insertions or deletions in red here, if it's not a multiple of 3, it will create a frame shift, the codons will be shifted, and so you create a random protein that will be non-functional. Okay, so it's a very easy assay. We can destroy genes one at a time. Destroying genes meaning we remove the presence of any functional protein. And so I knocked out a lot of Hox genes that are important for the development of enteroposteroid identity in those crustaceans. Just one quick example. Normally the mouth parts are in gray here on this electronic microscopy scan. There are little articulated parts for food processing in the mouth. And when you remove the gene called sexcom reduced, a famous Hox gene expressed in this area, you have transformation of those limb appendages. One of the limbs is transformed in an antenna. Another gene is transformed into a claw. So it's a Frankenstein experiment. It's quite spectacular. We can shift the identity of those legs by tweaking the Hox codes. And if I can summarize very quickly what we did, here is data that was generated by my colleague when I was there as a postdoc with Nipam Patel. You have the anteroposterior axis of those crustaceans with a schematic representation of all the leg types. Okay, and each leg type corresponds to a domain where you have the expression of 1 or 2 Hox genes. All right, and let me show you how we tested that model, this idea that 1 or 2 Hox genes are necessary to specify what kind of appendage you develop. So in the abdomen, for instance, you have 2 types of legs. We have the little swimmerets and we have the little anchors. Swimmerets anteriorly and anchors posteriorly. The swimmerets correspond to an area where you have 2 Hox genes expressed. You have abdominal B in yellow and abdominal A in magenta. So now what happens if we remove the magenta gene, if we remove abdominal A? The prediction is that only the yellow gene abdominal B will remain, and we should get only anchors, which is exactly what we obtained. All right, so very specific phenotypes validating this model. Then if we move slightly more anteriorly in the thorax, so the median region of this organism, normally in the WT means wild type, the regular individual without genetic modification, we have 2 types of legs. We have legs for walking forward and we have legs for jumping backward because they can jump when they're outside of the water, like beach hoppers. That's why they have those large hips with a lot of muscle in it. You can see it here. Right, so if we remove the magenta gene abdominal A, only ultra bithorax should remain, and we should modify the identity of those legs. And it's exactly what we get. So when we remove abdominal A, we get a very strange animal that has only forward walking legs. The reverse jumping legs were transformed into forward walking legs. That was really cool to me as someone who is interested in the mechanisms of evolution, because those parallel organisms are part of the crustacean group called Amphipoda, which literally means they have 2 kinds of legs or both kinds of legs. And their sister lineage, Isopoda, doesn't have this feature. They're called Isopoda because they have 1 type of legs, same type of legs. And with this knockout, with the removal of this protein, which was kind of brute force experiment because we removed the protein everywhere. So I don't think evolution works this way, but we pretty much generated from the perspective of leg morphology an isopod. All right, so it's, you know, scratching the surface of the iceberg, but that's the idea. With CRISPR, we can tweak development, remove genes, and get phenotypes that are of evolutionary interest. So then we can get deeper into it. I also want to mention that on top of this brute force removal of genes, I was successful doing one experiment of actual genome editing where you add something. I inserted the green fluorescence protein coding gene in frame with the Hox gene, in frame with the Hox gene antennapedia, which is expressed in the claws. So what you have in this schematic, it's on top here, you have the endogenous locus. So you have the regular antennapedia gene with its start codon ATG over here, and you have the first exon, and then you have an intron, and you're supposed to have splicing of exon 1, more on the right of the screen, right? And I co-injected my Cas9 and guide RNA over here, right at the start codon or almost, with a plasmid that is a donor template. So once you cleave the genome, instead of letting it repair itself without any template, you provide a plasmid that has homologous recombination arms on the side, those HR gray bars of about 1 kilobase on each side. And in the middle, we cheat because we are trying to force the insertion of a transgene, which was the jellyfish GFP, supposed to make green fluorescence protein and green fluorescence when you excite it the right way. And that worked. So we generated an animal that whenever it was generating, expressing antennapedia, it was also making green fluorescence, which was really exciting. And just for people who want to try this type of experiment, I have two cautionary tales to tell you. Make sure you beware of genetic variation, because the homologous repair here, it needs to come from the stock you inject. If you're using synthetic DNA that you made based on the genome sequence that was made somewhere else, you may not have the same genetic information allowing recombination, right? And also, depending on the endogenous expression level of your target gene, fluorescence can be extremely weak. So if you're screening with your eyes, you need to be very careful about detecting fluorescence. So maybe you use a sensitive camera instead and be in the total dark without light pollution from anything, including screens. All right, so now I'm going to shift to butterflies, but I need to tell you about my personal story at the risk to sound a little bit self-absorbed. So in 2004, I was an undergrad in biology. I was generally interested in evolution, neurobiology, biochemistry as well, a little bit. But I was also interested in literature, and I was reading novels by Vladimir Nabokov, who on top of being a very talented writer was also a lepidopterist, so a student and researcher of moths and butterflies. He worked at the Cornell University Museum, he published on butterflies, and he has a lot of butterfly biology in his novels. And so I was interested in writing as well, and maybe inspired by Nabokov. One day I wrote a short story that was science fiction. And to give you this story very quickly, it's in a dystopic future, a little bit like in the Black Mirrors series. So very close to our reality, but with technologies that have a dystopic effect on society. And in this future world, you have a biotech company that engineers butterflies and reproduce famous paintings on their wings. So this is a Photoshop. This does not exist, of course. So you have the Mona Lisa butterfly, you have a Picasso butterfly, and this biotech company sells those beauties to wealthy collectors, all right, for money, for profit. And so I was interested in this as an undergrad. I thought it was quite cool to have this reflection on our right to modify nature, to patent nature, what are the boundaries of modification of nature. It was an interesting story. And there's actually a whistleblower, a short story that works for this biotech, and he makes the butterflies free in nature. And then they are free to hybridize, and they create new creations, such as this mix of Mona Lisa and Picasso, right? So you have art and nature interacting. It's kind of cool. So it gets me into the biology of butterflies as an undergrad, pretty much. First of all, I was wondering, if we could, would we be able to engineer butterflies like this, create any kind of shapes with a genetic pencil? So I don't think we necessarily should, but it's a legit question, right? And then eventually I get into the basic question, how butterflies make shapes, stripes, colors, naturally. And it's by a twist of irony of existence, 20 years later, I'm here talking about our use of CRISPR in butterflies, which started to work around 2014 in my hands and the ones of other postdocs around the world. So we can inject the eggs of butterflies, and unlike the crustaceans, they have those dividing nuclei. They are a bag of nuclei. And you can inject this bag of nuclei, hoping that CRISPR will enter some of those nuclei and mutate your gene of interest. And that creates an animal that is a mosaic knockout. So why a mosaic? It's because it's not knocked out everywhere in its cells, only in a fraction of them, which allows the butterfly to be actually quite healthy during 1 month of embryonic and larval development and survive as an adult, even if the gene is important for its health. So as an example here, if you look carefully at this mutant butterfly, you have patches of the wings that are darker. It's because we knocked out a melanin inhibitor gene called ebony, and just like in Drosophila, when you remove that gene, you have darker melanin. So I will show you a lot of butterflies that have those kind of, we call them mutated clones, because they are cell lineages that start from the base of the wing and by mitosis propagate along the wing with those elongated shapes. And basically, there are those patches of cells that lack the gene compared to the rest of the butterfly. Okay, so I'm interested in how do we generate morphological diversity? There are 20,000 species of butterflies. You know a few of them, but most of them are in the tropics. And just the diversity of shapes and colors you can encounter in butterflies is just staggering. It's a crucible of evolutionary wizardry, and butterflies are pretty decent for genomics and for working in the lab. So how does DNA deploy patterns, shapes, pigments, scale types during development, and how does it evolve? So back in 2007, 2008, when I started my PhD with Bob Reed at the University of California, Irvine, we continued, I would say, an experiment done by colleagues mapping the genetic basis of shape variation in Heliconis butterflies. Those are tropical butterflies that have really a wide variety of patterns. So ignore the presence absence of red and just look at the shape of this median yellow band. It's central and continuous in this species, and in a sister species you can cross, it's wider and it's broken in the middle. And so we did crosses, we genotyped the offspring. I don't want to spend too much time on what is genetic mapping, but eventually we found a genetic region in the genome where we have a 0 recombinant interval. That means that in this region, you have the little mutations, maybe one mutation, maybe several mutations, that create 100% of this difference, continuous band versus broken band. And I got very excited because right in this small interval, there was a glowing candidate called Wnt. Wnt is a ligand, so it's a signaling molecule of the Wnt family, which is a very, very famous and I would say low-hanging fruit for developmental biologists. It is involved in the patterning of many aspects of our anatomy and in insects as well. And it was known to make pigmentation patterns before, like in some spotted fruit flies. Or, sorry, not Wnt, but related molecules like Wnt1, the famous wingless. So I was really thinking Wnt is a gene causing this difference in shapes, right? And so to explain to you what was the next step before CRISPR, I dissected caterpillars. So this animal is euthanized, so it's not alive. Please be reassured. And I'm making an incision on the side of this larva. It is before it's a butterfly and before metamorphosis. And above the legs, inside the thorax, we have those baby wing structures, which is absolutely amazing, right? So inside the larvae, you don't have some kind of juice or soup that becomes wings later. You already have tissues that are the future wings, and we call them the wing imaginal discs. You can see their overall shape, and you can see the veins that will give the strength to the wing later. So I can dissect those wing imaginal discs, and I can do experiments like in situ hybridizations to detect messenger RNA of a given gene. And so in this butterfly, here what you see in purple blue is where the gene is expressed, and it's basically expressed all around the yellow band, where you have black, all around the yellow band. In the other butterfly, which we used in our crosses, it's a completely switch expression pattern, and it's rather strongly expressed in the middle, exactly where you have melanic areas inside the broken band. Excuse me. So we think that at this step, we have a very, very good correlation between Wnt expression, which changes radically between the two genotypes, and the presence of boundaries between light and dark patterns. So it seems like we nailed the pencil that draws wing patterns pretty well. At that time, we didn't have CRISPR. However, we had a drug called heparin, which independently of its mechanisms in humans as a blood thinner, completely different mechanism, it interacts with wingless or wind signaling. It was known from this paper. So wind signals are exported outside of cells here, and they're going to go to a receiver cells to the right receptors, and they need to be protected or coated by heparan sulfite glycosaminoglycans like heparin. And if you add heparin to the cells, to the outside of the cells, wind signaling is expected to be stronger, faster, better. So in those butterflies, is an example, Wnt is expressed in the dark areas. So over here and over here, this is, two stripes in the larval stage that corresponds to those melanic areas in the adult. And when I injected heparin, I was expecting those melanic areas to expand. And this is exactly what occurred, right? So that was a gain of function. It was a quick and dirty way to enhance Wnt signaling and to test, if the phenotypes would have the right direction, meaning Wnt expressed in the melanic areas, if you give them a boost, they start to cover the yellow areas. You expand the size of those patterns until you have a completely dark butterfly. So that was cool, but it's years later that we were able to remove Wnt in a lot of butterflies. And as predicted, we had the completely opposite phenotype. We removed Wnt from those presentive melanic areas, this one over here and this one over here. And we created a butterfly that has no Wnt, and as a matter of fact, that this very expanded yellow pattern. And it works really well with this gene. We really love this gene. We knocked out Wnt in a bunch of butterflies, 7 species for the first papers, and then a lot more, and we're still doing this sort of comparative approach. So if you take the wing disc out of this beautiful butterfly, Agrolis vanille, you get a completely different expression of Wnt, those little dots. And when you remove Wnt, you get the removal of those of the silver patterns comparing to, that are made by those dots. So for instance, you can have 3 dots here, they make those silver patterns with a little bit of dark on the outside. When you knock out Wnt, this is completely gone, all right, because you remove the functional protein during larval development, we suspect. In the hind wing, it's even better. We have dot, dot, a finger of expression, dot, dot, dot. The wild type pattern in the hind wing corresponds to those silver spots with the same shape. You remove Wnt, you get a complete absence of them. There's a little bit of residual activity, we think, from another Wnt ligand, probably Wnt 1 and 6. The reverse experiment with the heparin works also pretty well. So you can enhance Wnt signaling. You expect an increasing of those patterns. And this is what happens. So this is wild type. You just inject water, you get the regular patterns. If you increase Wnt signaling, those silver spots around here gets bigger and bigger. So all the elements that I highlighted over here that are expressing Wnt gets larger with heparin. So now with this assay and CRISPR, we can increase gene expression or gene activity and remove it with CRISPR. What about the monarchs? Everybody loves the monarchs, right? So Angie and Darly, graduate students at that time, now they're both postdocs, looked at the expression of Wnt and found this completely different expression that seems to line up the veins, line up the wing veins. And when we knocked out Wnt in the butterflies, we were expecting a removal of the black areas that overlay with the veins of the monarch. But we get something quite, slightly different that I will explain with a magnifying version. So this is what you get around the veins. You get veins, you get an outline of black, and then you get the orange. And that's allowing the butterflies to make a lot of contrast, right? Orange and black, very, very strong contrast, is to signal their toxicity to bird predators. But if you might know the trick in visual design, if you want to make even more contrast, use a little outline of white, right? So you see this little outline of white that provides even more contrast. And Winte is expressed around this area where you have the white outline. When we remove Winte, this outline is derepressed, it's out of control, and it takes over the whole wing. So that was an example of tinkering, right? Winte did a lot of patterns during butterfly evolution, and in the monarchs, it's a clear example of tinkering. It changed its expression to be around the veins, and it's been involved in making the right amount of white outline, pretty much. If you remove it, you get too much. So we knocked out Winte in a lot of butterflies thanks to many talented junior scientists, and this is published if you want to check out the paper. So I want to talk a little bit about the painted ladies here and still about Winte. So Winte is clearly involved in pattern induction. So in this more complicated butterfly, Winte is expressed in the center of the wing, and this is the wild type pattern here, where you have those concentric waves. And it's not expressed in the eye spots on the right side, and it's not expressed on the more left side. And when you remove Winte, you get a complete removal of the patterns, the stripes or waves that are exactly where Winte is expressed. So it really shows causality. Winte is necessary to make those patterns. Again, wild type and after CRISPR. And even maybe as well interesting, on the outside of the wings, we have an effect that is a little bit harder to see. But if you look at the expression of Winte in the imaginal disk, we have those central areas here, central stripes, and you have something more on the outside. So let's zoom on this area. So in the wild-type butterfly, you have eye spots, and a little bit closer to the wing margin, you have those blue chevron patterns here. And when you remove Winte, you don't change the eye spots, but you change a little bit the chevrons. So I'm going to make it easier for you to see. In the wild-type side, if you look at the eye spots and chevron, it looks a little bit like a minion that is happy minion. In the mutant, it's a sad minion. All right, everybody sees that, right? Happy minion, sad minion. We were interested in the question, how Winte is working. Is it working like most other Winte ligands, or is it one of those strange Winte ligands? And most Winte ligands needs to be secreted in the source cells. They need to be acylated by a gene called porcupine, and then they need to be cargoed through the Golgi and endoplasmic reticulum for maturation, thanks to the gene Wintless. So we knocked out porcupine and Wintless, and we were curious about what we would get. In fact, we got a perfect replication of the sad minion phenotype, strongly implying that Winte is using the typical traditional pathway for secretion. It's not a weird Winte ligand. So we are really enjoying this butterfly in the lab. They're easy to rear, and we're really enjoying this phenotype because it has those very specific effects that are independent of the function of other Winte signals. So we're going to move forward dissecting what is more downstream, what are the receptors, what is the signal transduction of Winte, and what are the target genes. So how Winte makes shapes. Moving on, we knocked out other genes in butterflies, and just want you to get just a flavor of what's going on here. So we have mutant clones, those orange patches here elongating over the blue chevron of the hind wing is a mutant clone for a gene. I mentioned this type of gene before. It's a Hox gene. So it's a gene that is important in determining what along the anteroposterior axis. And so you can see in the mutant, the blue chevrons from the hind wing is interrupted by melanic patterns that are on the forewing. The forewing is the anterior wing, the hind wing is the posterior wing. So how do we make those kind of copy-paste effects between cell identities, right? And how butterflies differentiate their anterior wing from their posterior wing? Well, very clearly they use the Hox gene ultra bithorax, like in Drosophila. So when we knock out this gene, we get high lethality, but we get little patches of wings on the hind wing that take the forewing identity. So you can see this copy paste of orange and salmon colors onto the hind wing. So if you look on the left, the hind wing is supposed to be beige and hairy, and in the mutant, you copy paste pink scales from the forewing. We also replicated those results in other species, including the mealmoth, which I'm very excited about. So the Indian mealmoth Plodia interpunctella is behaving very well in our lab. We also have an NSF grant to develop CRISPR methodologies, and don't hesitate to contact us for protocols or for learning more. When we knock out UBX, we get transformation of the hind wings into forewings. So the hind wings of those moths are usually hidden and they work like an unpigmented fan. When the moth is at rest, it's a little bit like this. The hind wings are completely folded. But the forewings are the actual wings used for flight, so they are much longer and they're more tough, thicker, and they don't fold. When we knock out UBX, we get an intermediate morphology, really showing that UBX is involved in making the hind wing specialized compared to the forewings. So CRISPR works really well for perturbation assays in these moths. We have a pretty good setup for rearing and for injections, and it's very cheap as well to work with. I need to briefly mention that we have regular transgenesis based on transposons to work. So this is a moth that is expressing GFP in the eyes using a piggyback modified vector. So that works. You can ask me if you're interested. But next, we are interested in working on CRISPR in the system. So they're very easy to maintain, they're resistant to inbreeding, they're a worldwide pest of stored food. So I think they're a pretty cool model system for Lepidoptera, not just butterflies, but also moths. With the help of Paul Shirk, we acquired a mutant strain that has white eyes. And we mapped the mutation responsible for those white eyes to a very small region that we used genetic crosses for that. The region was interesting because it contained 3 candidate genes involved in eye pigmentation. They're named after their name their name and function in Drosophila: the white gene, the scarlet gene, and the cinnabar gene. And so we compared the wild type strain to the mutant strain, and we found that the mutant strain had a coding mutation, just 1 deletion in an exon of the white eye gene, creating a frame shift. So that was very cool. 1 base pair mutated in the wild type strain. Maybe we can try to fix it with CRISPR. So we have the target sequence of the mutant right here. We have the guide RNA to go after this area, and we co-inject it with a short donor template called the SSODN. It's a single-stranded DNA that has short homology arms on the side for recombination, for homologous repair. And in the middle, it has 2 things. It has the fix. So we put back the deleted cytosine that will repair the mutation. And we also have an additional mutation to prevent secondary cut by Cas9. And that works. We have a very highly efficient method for CRISPR insertions. 1/3 of the injected moths successfully repaired the mutation in some of their eye tissue. So when it does not work, you have a brown eye. But when it works a little bit, some of the eye tissues are going to recover some brown pigmentation. So it can be diffuse, or some clones can make those elegant stripes. But some moths work really well. We call them bilateral. They recover pigmentation on both eyes. So almost all of this eye, and a pretty good recovery in the other eye. So that works really well, and we are starting to put methods on my website done by Krista, who also did a lot of this Plodia work, if you're interested. So next, I would like to mention briefly our work on iridescent. A lot of butterflies have very cool nanostructures on their wings that create very cool iridescent color, so angle-dependent metallic reflections. But flying under the radar, I would say, in our backyards, we have this Coleus butterfly that is also iridescent. But you cannot really see it because this iridescence is ultraviolet. You need a special camera to image the iridescence of those butterflies, which is really, really exciting because those butterflies are right here. They're a very common butterfly called the sulfur butterfly, while iridescent butterflies that are blue or green or orange tend to be in the tropics. You know, they're a little bit less common around us. So we got an NSF grant led by Brian Canterman at Auburn University, and a lot of help on the genomics from Chris Sweet in Sweden and Wardwa, a biologist who worked with those Coleus butterflies for most of his career. And here's a video I made of a Coleus butterfly. You can see the iridescence is on the dorsal side. This iridescence is driven by very, very cool nanostructures on the surface of their scales. Those are amazing. It's very hard to understand how butterflies can make this with a chitin material. We're starting to work on this, but I won't have much to tell about it. But long story short, we actually have two species. There is one that is iridescent and another that is not. For the little story, only males are iridescent because this is a color that is used in sexual selection. So males are using the flashes of UV iridescence to signal to the females that they are the right species. We have a cool system here for speciation genomics because the iridescent butterfly, the orange one, used to be only west of the Mississippi River. And during the 19th century and the 20th century, what happened is that people started to plant alfalfa, which is a very good host for this butterfly. Alfalfa, because it's used as feed for cattle, right? Also, the landscape changed a lot of forest was cleared. So progressively, starting from the middle of the 19th century, those butterflies started to expand westward and then northward. And now they overlap with another butterfly that does not have the UV iridescence, Coleus philodecine. And those things, those two species now occur at very high densities in the alfalfa fields of the East Coast, sorry. So we go to an alfalfa farm in Maryland, so next to DC. We know those two butterflies got reunited in 1927. It's very well dated and documented. So for about 80, 90 years, those two butterflies that were separated, but sister species, got reunited. It turns out they can hybridize and exchange most of their genome. However, they have a lot of differences on their sex chromosome. It's called the Z chromosome. It's the equivalent of our X chromosome because the heterogametic sex of butterflies is the female, not the male. So anyway, we use genetic mapping thanks to hybrid crosses that had been done 20 years ago by Adam Porter and his grad student Bai-Ching Wang. We genotyped all those butterflies, the UV iridescent versus the non-UV iridescence, using next generation sequencing called 2B-RAD sequencing in this case to make it cheap. And we narrowed the genetic interval here called U locus to a small region that contains a dozen of genes. And again, so in this region, we know that there is a mutation or some mutations that underlie the presence absence of UV iridescence. Sorry. And we were very excited because on one side of this interval, there is a gene called bric-a-brac or BAB, which is already known to control male-specific structures in Drosophila. So it controls the male-specific pigmentation of the abdomen in fruit flies. You know, Drosophila melanogaster is called melanogaster because males have a black belly, melanogaster, a black belly. That's done by BAB. BAB is a key component of this pattern regulation. And Drosophila fruit flies also have structures on their forelegs involved in copulation called the sex comb. Those are modified bristles. They're only in the males. And basically in Drosophila, when you remove BAB, you get more of the male structures. So that means that BAB is a repressor of male traits in Drosophila. And again, here in Coleus, we had a male-specific trait, the UV iridescence, with BAB as a candidate. So likely the gene that is a repressor of the male-specific trait, the UV iridescence. So we use CRISPR to knock out BAB. Here's the ventral side of the butterfly where you're not supposed to have UV iridescence, right? So you have the wild type, UV reflectance here, it's rather opaque. You do not have UV iridescence except on a small metallic spot. When you remove BAB, it's supposed to be a repressor of the male traits. We get, and this is amazing, ectopic UV iridescence covering the entire butterfly. So we made those butterflies iridescent everywhere. When we knocked out the gene in the females, here you have the dorsal side, the females are not supposed to be iridescent. You have the wild type on the left. Same thing. BAB is actually expressed everywhere in these wings, but it's repressing UV iridescence. So when you remove it, when you remove any kind of BAB functional protein, the UV state is de-repressed. So it's turned on and you get UV iridescent clones, those little mutant patches, right? Same thing in the males of the other species, the one that has always been on the East Coast, but it's been always been not iridescent. All right, the wild type is not UV iridescent. If you knock out BAB, you have those UV iridescent patterns that show up. We can zoom in on those scales. We can compare the patches that are mutant with the ectopic scales with the parts that are still opaque. And we can overlay UV photography, regular photography, and electron microscopy to really show that every single scale in the mutant patches, both the scales on the top of the layer and on the underside of the layer, the green ones, both are transformed. So all the scales are supposed to express BAB and repress UV states. And when you remove BAB, you get conversion into UV iridescence everywhere, which is really cool. So long story short, BAB knockouts create a gain of iridescence regardless of the wing surface, ventral or dorsal, the sex, male or female, or the species. So BAB is a repressor of UV scales. If you remove BAB, you get UV scales back in. So what have we learned so far with CRISPR in butterflies? I showed you quickly that wind signaling is important and instructive for pattern formation, the formation of shapes like dots, spots, stripes. UBX is important for differentiating the hind wing for the forewings, which is very common in butterflies. And we have bric-a-brac in a specific lineage of butterflies that underlies structural colors. And also knockouts or important gene perturbation assays. We're trying to make it actual genome editing with precision editing in the moth, the male moth Plodia. So I'm going to take very quickly 2 minutes to mention, I've also been doing CRISPR with undergrads. So this is a classroom with undergrads, usually 12 or 15, who are doing CRISPR injections and knockouts as part of their curriculum. It's a genome editing laboratory course. I have a lot of experience with this. I've done it for several years. And those students are having a blast. They learn about genetics, they learn about genome editing, and we talk a lot about bioethics as well. So the painted ladies can create a really scalable amount of eggs. You can get hundreds of eggs easily in the classroom in one afternoon for injections. You can order painted ladies online. They're so easy to rear that they're sold as a toy to kids. Even a 4-year-old can grow them. So they're really ideal, and they're gigantic eggs that you can inject with cheap injection material. So you can rear the larvae on artificial diet. You just need a lot of cups and an environment at a stable temperature. But you can make mutant butterflies with your students. And we published a little paper on this. I will mention it again. So butterflies have really gigantic eggs, larger than most fish eggs. Than laboratory fishes, let's say, and almost as large as xenopus frogs. We also did xenopus frog injections. It's a little bit more involved to get a lot of eggs regularly over several weeks. So butterflies are really the sweet spot, gigantic eggs that are easily to inject for sheep. If you're trying other organisms like Drosophila, mosquitoes, or I don't know, snails or marine organisms, the eggs are going to be smaller. The eggs might be easy to get, like sea urchins can produce a lot of eggs on demand, but it's going to be so small that you're going to need very, very talented undergrads to inject them, and that requires expensive microinjectors with high precision. With butterflies, we can use super cheap equipment, so you can have, 2, 3, 4, 5 stations. And if you have TAs, you can crank it to 12 students, 24 students, 36 students in the classroom. I'm happy to talk in private about the logistics of this. So we use those very simple injectors. We use capillaries that are easy to load, capillary glass needles that are easy to load and easy to pull, very cheap. If you don't have pressurized air in your room, you don't need compressed gas. You just need an air compressor, which is really dirt cheap. So you can really do this anywhere in the world. You need a -80 to store your guide RNAs, and that's about it. So that's really cool. Undergrads have successfully replicated phenotypes, like including ones that are not published, like this Frizzle2 receptor phenotype for winter. And this class is exportable. I worked to help Linky. She's at Stetson University in a primarily undergraduate teaching position. And her students have done mind-blowing phenotypes already in the painted ladies, such as this optics knockout. It's crazy, right? Which makes completely dark butterflies. It's a publication from the Reed Lab from a few years ago. They reproduce that. And imagine being an undergrad and doing this. This is really interesting. I think it's life-changing for them. So if you have any question on all of this, please contact me on Twitter or by email. And if you're interested in the class design, check out this paper. I want to thank all my collaborators and lab members, and thank you for your attention. I'll take any question.
Okay, so if anybody has any questions, you can either raise your hand and we will call on you to ask them, or you can post them within the Q&A section in your Zoom. So in our Zoom right now from North Carolina State, somebody is asking, what are the possible ecological effects of these edits, and do they interfere with any reproductive isolations?
Yes, so I'm going to go back quickly to maybe, okay, so this slide, I'm just pulling it out of the top hat right now. So on this lower corner here, you have a courtship attempt from a UV iridescent butterfly that is courting a female thanks to its UV patterns. We could imagine that the BAB knockouts that I showed, they're going to make the other species UV iridescent. So that will make those males more attractive to the females of the other species. So that's one example. I don't think we should, it's something that is worth studying. Maybe we could check if those edits manage to improve the mating success in a laboratory setup. That could be interesting. But because they're dirty knockouts, because we remove the protein, I would not be surprised if we change the behavior of those butterflies, or if those knockouts have some kind of negative effects on other parts of the biology, because the genes we tend to target are highly pleiotropic. They tend to have a lot of, so not off-target effects, but unwanted effects besides the wings. I don't know if it really answers your question. If you want to specify, if you had something in mind, please feel free to check in again.
Okay, we have another question asking about pet animals. Do you think there's any application in order to edit the pet animals like a dog or a cat in order to change the color or any kind of visual components?
Yeah, so I definitely think CRISPR could be used for making designer pets, cats or dogs. or the object of study from the perspective of color genetics. So now, do we really want to make designer pets with CRISPR modified patterns? I don't know, I feel a little bit weird about it. However, because we domesticated pets using artificial selection, we also selected for genetic diseases, and it's definitely possible to maybe fix those mutations and make our dogs healthier with genome editing. Some people are working on this. Regarding designer pets from the perspective of color pattern, I would not be surprised if you start to see CRISPR-edited aquarium stuff like zebrafish or other fishes in the continuation of the famous glow fishes that are fluorescent or phosphorescent fishes that are already commercialized. Do I want to live in a world where we sell genetically modified pets? I'm not really sure, but it's definitely a possibility.
Okay, and we have another question from Mason.
Hi, Dr. Martin. That was a really great presentation. I wanted to ask that if, in addition to the question about reproductive isolation, that there could be some really cool ecological and conservation applications of being able to manipulate the coloration of the butterfly. So I was wondering, from what you've looked at so far, if you've thought of any applications of using these different coloration pathways for conservation purposes, such as perhaps doing some type of artificial Mullerian mimicry or something like that that would help species that might be endangered evade predation or something to that effect.
Yeah, that's a good question. I mean, I love this kind of forward-looking thinking. What can we do with genome editing? So I'd say I am skeptical about. It's strange. So I'm skeptical about the use of genome editing of the color patterns to have any kind of conservation benefits, because let's pretend we make a mutant butterfly that has a better traits, a better color pattern, or it's healthier and something like this. We're gonna make this butterfly in one genetic stock, and you will have to release really a lot of butterflies in the population so they can propagate their better genes. So people have thought about using maybe gene drives in mosquitoes, so more sophisticated systems that can cheat with gene propagation to make genes invade the population. So when you think about it, you could make mosquitoes resistant to the malaria parasite, and if you could spread that in a population, those genes could expand using a gene drive. I'm really hand wavy right now. So I have not thought about it seriously from the perspective of wing patterns. I'd say right now we're just interested in the fundamental questions of what genes or how genes are making shapes and colors and how it evolves. So I'm interested in the ecological relevance of those patterns in terms of application. I haven't thought about it too much, maybe to be honest. So that's my long answer to you.
Okay, great. Thank you very much.
Thank you.
Yes, I also have a question as well. You said that you were able to insert a GFP using homology-directed repair. What's the efficiency level of homology-directed repair within butterfly compared to non-homologous end joining?
That's a good question. That's an excellent question. And we have not done the proper experiments to compare, right? So in the crustacean, the knockouts, so no repair template, just the knockout of the host genes was extremely efficient. And I think because they have this slow cell division, you can make it 100% if you inject 2 guide RNAs, blah, blah, blah. The repair, the knock-in of the GFP was very low efficiency. I got one batch with a 4% efficiency rate 4% of the offspring was fluorescent. So it's pretty low. And it depends on the day and the needles and the mood of the injector you can fare. In butterflies, so in moths, we repair the white eye mutation to make it pigmented again. That I'm very excited because it was very efficient in our assay. A third of the moths were edited. Now the question, Brendan, is going to be, can we make it as efficient, 25% or a third efficiency? When we try to, instead of tweaking one single nucleotide, when we try to insert larger transgenes like a GFP. It is thought that in a lot of organisms, the longer the gene you're trying to insert is, the longer you need to have homology repair arms on the side of your plasmid, of your donor plasmid. And then efficiency is going to drop no matter what. So we are in this space at the beginning, and I don't have a very good answer, but we expect that if you want to make a lot of modifications, it's going to be low efficiency, maybe of a few percents.
Okay. Does anybody have any other questions that they'd like for Dr. Martin?
Actually, I did have a follow-up question about that. In terms of the size of the constructs in the case of the homology-directed repair and looking at the efficiency, what are the kind of upper limits of the size of those vectors right now in terms of what you can deliver?
I heard of people being successful injecting plasmids that were pretty large, like 8 or 9 kilobases.
Okay.
So that makes it possible to insert a cassette of 2 to 3 kilobases with homology arms of 2 kilobases on the side. Mm-hmm. I have the feeling, it's kind of what people are trying to do. You have alternative to donor plasmids. You can also synthesize long single-stranded DNAs. You can also buy them. They're called megamers from the company IDTs. You can do it in your lab for cheaper as well. That works really well in mice, maybe in mammals. And yeah, so then can CRISPR insert super long stuff? Probably if you use back clones, back plasmids, you can have like super long homology arms. I'm sure you can engineer this type of stuff. It hasn't been very common, to my knowledge, in arthropods to go larger than those 8-9 kb plasmids.
Okay, great, thank you.
Okay, do we have any additional questions? Okay, let's see. Dr. McGill, you have a question you'd like to ask?
Yeah, you were injecting eggs in the butterflies, right?
We were injecting what? Sorry, I missed that.
Eggs, fertilized eggs? Or...
Fertilized eggs, absolutely.
And so have you looked at the inheritance of these in the future generations? Is it?
Yes, so we are trying to propagate those mutations in the germline. It's challenging when the gene is lethal. It's important for embryonic development. We have some success with Winte, which makes those beautiful, mutant wings like in stripes. And that works at pretty high efficiency, except that the females are sterile, so it's hard to breed those butterflies. The meal moth, the little Plodia meal moth, I think is really where we should push, because we can make a lot of inbred lines and we have no problem propagating both the knockout alleles of the pigmented eye and the repaired version of the pigmented eye. So in the Plodia moths, we have no problem creating the next generation. So in other words, that means when we inject the early embryos, we have access to the cells that are the germline already. The germline is not protected from whatever we inject, or not yet protected. We can inject early enough.
Interesting.
Okay, anybody else? Okay, in that case, thank you, Dr. Martin. We are going to be taking...